So you should have gotten a notification now. So if you don't want to appear in the video, basically don't say anything. So today we have Asif Equbal. He did his bachelor's and master's in chemistry in the Indian Institute of Science, Education and Research in Mohali, and a PhD in Aarhus in Denmark with Niels Christian Nielsen in NMR, solid state NMR. He did a short postdoc with Professor Mathur in Mumbai, if I got this correctly, and then since 2016, he's a postdoctoral researcher at the University of California in Santa Barbara in the group of Songi Han. And today he's talking about the role of EPR detection in deciphering DNP mechanism. And I'm gonna unmute you. I think you can unmute yourself, Asif, if I'm right. Yeah, so feel free. Stage is yours.
Okay, all right. Thank you, Nino, and thank you all the organizers for giving me this opportunity to talk about my research, which is on DNP development. I'm very excited about giving this talk because in last 3 years of my research, I have realized that DNP development goes hand in hand with EPR development. So, and this forum is an excellent forum to talk about DNP mechanism from the EPR perspective. So let me start with something very basic. We all know that NMR has the issue of sensitivity. The sensitivity is in NMR is intrinsically low, and it comes from the fact that we probe a very small energy gap in NMR. There are ways to improve the sensitivity. We can either increase the magnetic field or we can lower the temperature to increase the polarization, as you see in this formula. Let me switch to the pointer mode. Oh. Just give me a second. Okay. There is an alternative way to increase polarization, and that's called hyperpolarization via transfer from a more sensitive spin. And so that's something that leads to gives rise to DNP. The concept was already well known even in 1950s, but then it became applicable only in last 1 or 2 decades, thanks to Bob Griffin, who brought the renaissance with the development of gyrotron. And since then, there have been many applications of DNP. The reason in DNP, the source of polarization for the nuclear NMR is basically electron, and fundamentally electron spins and nuclear spins, they are very similar. There is difference, and that difference comes from the time scale of the two end of magnetic resonance. In case of NMR, the gyromagnetic ratio is typically in megahertz per Tesla, and that is something that makes NMR insensitive. On the other hand, in EPR, the gyromagnetic ratio is a factor of at least 1,000 more, which makes EPR very sensitive. As a result of that, the relaxation in EPR is also much faster, so one can repeat an EPR experiment much faster than one can do in case of NMR. The third major difference that also comes from the gyromagnetic ratio is coupling. In NMR, the coupling is small. It's typically in kilohertz or hundreds of hertz, which gives rise to excellent resolution in NMR, whereas in EPR, the coupling is large, which impedes the resolution. So ideally, in DNP, what we want to do is we want to combine the best of these two worlds. We want to have the sensitivity of EPR, and we want to be able to repeat an experiment much faster the way it's done in EPR, but we want to contain the resolution of NMR. So that's the aim with DNP, and if something like this is feasible, it would be great. Practically, for DNP to be feasible for a target molecule, we need a source of unpaired electron, and that source of unpaired electron is normally comes in the form of a stable radical that's dissolved in a glassing matrix, which also contains the target molecule that we want to study via NMR. To polarize the target to the nucleus in the target molecule, we have to use microwave irradiation, and that transfers the polarization from electron to the directly coupled nucleus, and then the enhanced polarization of the nuclear spin is spin diffused to the target sample. So that's how typically a DNP experiment from application point of view is used. There are multiple factors in DNP which makes this technique very interesting from research point of view. The important factors are relaxation, that in turn depends on many other factors. Then the microwave characteristic used for deriving the DNP is also very important. So the microwave power or frequency is important. The third factor is the coupling network between the electron spins. So in this presentation, I will basically focus on the third aspect of the third important parameter of the DNP. And that's because coupling network is important because it determines the kind of mechanism that would be driven in under a DNP condition. So suppose if you have a diluted electron spin, then either Overhauser effect or solid effect would be stringent, would be feasible if we have diluted unpaired electrons. Within this, in the case of Overhauser effect, to give rise to high enhancement, there has to be an efficient cross polarization between electrons and the nuclear and the coupled nuclear spin, which may not be possible in the case of insulating solids. So this mechanism is popular mostly in either in liquid sample or in conducting solids. Solid effect, on the other hand, is possible in all kinds of solids, but then it comes with a very harsh condition in terms of microwave irradiation. One has to drive or saturate the zero quantum or double quantum electron nuclear transition in order to have an efficient solid effect DNP. And achieving making this transition possible at higher magnetic field becomes infeasible, because when we go to higher magnetic field, the mixing of electron nuclear states become the probability lowers down, and also the microwave power that's available at higher magnetic field that's also is much lower. So a solid effect at high magnetic field is not a solution. The solution rather comes from the mechanisms that involve coupled electron spins. So these mechanisms can be broadly classified as triple flip mechanism, which requires at least 2 coupled electron spins, and they have to be coupled to a nuclear spin to which the polarization has to be transferred. The other condition is that the energy difference between the 2 electron spins that has to match the nuclear energy, so that the process basically it drives through a triple flip mechanism. So if the triple flip happens, the energy should be conserved, because this triple flip is very much intrinsic to the system. It does not depend on the microwave. So this is a fundamental mechanism that happens in coupled electron and nuclear spins, and that is used to drive the DNP at present condition. And this would also be the focus of my talk. So let's spend another 30 seconds on this aspect of DNP. There are 2 important parameters. The one is 2 factors that determine the efficiency of this process. The first is coupling between the electron spins, and coupling basically its role is to it determines the transfer rate. And in the scenario where relaxation is a competing process, we want the transfer rate to be high enough. So it's electron-electron coupling that determines the flip-flop process here. The other factor that determines the extent of transfer is the polarization difference between the 2 electrons. So here we ideally want to create a large polarization difference between the 2 coupled electron spins. And if we do that, then through the triple flip mechanism, the electrons, they want to reach a new kind of equilibrium. And in that process, the nuclear enhancement happens. So creating a large polarization gradient between the cross effect or thermal mixing effect satisfying electron is very important for the triple flip DNP mechanism to be efficient. So There are other factors like when it comes to coupling, there has to be a balance between dipolar and exchange coupling. I won't go into all of that. And even when we want to create a large polarization difference, the role of microwave power, the relaxation, and the intrinsic electron spectral diffusion, that all of it plays a role. So there are multiple factors there. So let's now, let me talk to you about the challenges that are there in DNP. I showed you initially this chart that in DNP, ideally we want to have very high sensitivity and we want to be able to repeat experiment faster and also containing resolution, but it comes with a condition. And the condition is basically at present, there the DNP experiments, they are efficient at lower magnetic field, at lower temperature, and also like at slow sample spinning. But these conditions are not the ideal conditions for an NMR experiment. So we want to rather push the efficiency of a boundary of DNP to more practically relevant condition for the NMR. So that's like a challenge in DNP that many people are trying to solve. So the way forward is to solve this challenge is to first identify the problem, and to identify the problem, we need to understand the mechanism, identify the mechanisms. And once we know what kind of mechanism is operating, then we can have a specific solution catered to the specific mechanism. So people normally try to identify the DNP mechanism using NMR experiment. They record basically DNP enhancement as a function of either microwave frequency or as a function of microwave power. And then they get certain profile, which is fitted with some known theory based on many approximation. And one can, in principle, know what kind of mechanism is more dominant there, but then it requires a lot of background information. However, even in these cases, the key information like the coupling network or the polarization gradient that would be generated under microwave irradiation, those are very essential informations to know the efficiency of DNP. Those informations cannot be obtained using NMR measurement. NMR in the end will tell you like how much of enhancement is there. So we do not really know how that enhancement, we reach to that enhancement. All of us would agree that since the source of polarization is electron, so having an handle on knowing the electron spin dynamics would the best way to identify the DNP mechanism. So this is something we have been trying in our lab to do, and also like other people, for instance, people in Weizmann, Daniela Goldfarb and Shimon Vega, they have used EPR detection to understand the DNP mechanism better. Others have not been able to contribute much to it because the kind of setup that's used for DNP is the commercial setup basically does not have the capability of doing EPR detection, and that's because the microwave source that's used, it's not tunable. First of all, it does not have a pulse shaping capability, and it's also not, the source is also not phase sensitive, and all of these criteria are needed for doing an EPR detection along with NMR experiment. We luckily have that feature with our home built setup that several PhD and postdoc in the lab of Songi Han have contributed to in the last 5 to 10 years. And in my talk, I'll be basically showing you some of the results that we got that in last few months, I will say, which helped us to understand DNP mechanisms better. So to be more specific, to highlight how EPR detection in particular can help to understand DNP mechanism, I'm showing you a cartoon here. So under a practical considerations, where the way DNP experiment is done, we take certain radical in high concentration. The concentration is typically much higher than what one uses for EPR experiment. So we use normally like around 20 to 40 millimolar, and sometimes it can even be higher, like 60, 70 millimolar of radical. When we have such a highly concentrated radical dispersed in a glassing matrix, we do not really know about the distribution of electron spins, whether the electron spins are all distributed homogeneously across the sample, or whether they have the tendency to interact with each other or interact with the solvent. So the interaction of radical interaction or radical solvent interaction, that's something which cannot be predicted, and that's influences the DNP mechanism quite a lot. So if all the radicals, they tend to have interact more with each other, then a thermal mixing mechanism would be more operative. If they are remain isolated, then solid effect would be the more efficient, would be the more likely process. So the coupling network really determines the DNP mechanism. So ideally, one can do some simple EPR experiment and know about the dynamics and the other parameters about electron spins. One can do an echo-detected EPR experiment, which can help us to know about the distribution of electron spins as a function of frequency, resonance frequency. And since it's an echo-detected experiment, it would also be weighted by relaxation. And that's very important, because effectively those spins which can be perturbed with the given microwave, they have a more significant role in the DNP process. So knowing the EPR line shape is very important to understand DNP mechanism. The other thing is to know the relaxation rate of the electron spins, and also to know for which we can do a simple T1E measurement, like either inversion recovery or saturation recovery. And then one also needs to know about the shape of electron spin polarization, for which an experiment like ELDOR, which is a pump probe experiment, can be very handy. So these experiments can be used for developing mechanisms. They can also be used for developing polarizing agent. And I'll just give you one example, because this seems to be a very important example. So in 2013, Lyndon Amsley and which also included Gunnar Jaschke, they developed a series of radicals. And the idea was like, if a radical is bulkier and has a rigid framework, then its relaxation would be slower. So a bunch of radicals were synthesized, and the DNP enhancement were correlated with the relaxation rate. And clearly, such study, it led to the best performing DNP radical, which is Steckpol. You can see number 6 here is Steckpol, which has long T1E and also like high enhancement. There are other experiments like ENDOR and eSIM, which can be done to know about hyperfine coupling. That's also very important from DNP aspect. I won't go into that. I'll rather jump to the main part of my talk, which is to understand the DNP mechanism using EPR detection. I'll skip any question now, but I'll take it later on. So in this talk, I'll be telling you about a story that was generated using trital radical. So I'm sure all of you know about trital radical. It's a narrow line radical, monoradical, and its G anisotropy is much smaller than proton Larmor frequency. So suppose we have 5 millimolar concentrated trital dissolved in a glassing matrix, and if we assume that all the tritals, they are segregated, and then ideally the only mechanism that would be operative would be solid effect, which means we will get DNP enhancement when we do a microwave irradiation at a frequency which is approximately nuclear Larmor frequency, or in our case 300 megahertz apart from the trital center. So we get either zero quantum solid effect or double quantum solid effect. What would happen when we go to higher concentration? So that can be a question. And if one has to predict just based on NMR experiment, one would say that, okay, if you increase the concentration, then the solid effect enhancement would initially will increase, and after reaching an optimum, it will start lowering down, and there won't be any other enhancement that would appear in case of trital, because trital is very narrow line radical. So at very high concentration, what would happen is the relaxation would become very fast, and there won't be any enhancement at all. So that's something that we thought would happen, but then we were very surprised when we did a more systematic study. So here I'll be showing you about DNP profile for trital as a function of concentration. So Going from 5 millimolar to 10 millimolar, we see like there is definitely solid effect, but a small feature is there in the center. And when you further increase the concentration of tritol, that feature that's there in the center of the tritol, it becomes very prominent. And if we see the enhancement at those conditions, those enhancements are much higher than what one gets from solid effect. So solid effect initially increases and then it quenches because of fast relaxation. But the thermal mixing DNP, it becomes very prominent. And this is something which was completely unexpected. So we uh, we wanted to, so basically what happened was the trityl radicals, which were initially all isolated and diluted in the glassing matrix, they clustered and they gave rise to thermal mixing. So this is our hypothesis, but how do we prove that? We can do that using EPR based detection. So already there were some theoretical predictions made for the thermal mixing, and that served as a good guideline for us to probe this mechanism via DNP. So first prediction was that there would be an asymmetry in the EPR line shape if thermal mixing has to operate, and that prediction came from a very rigorous theory by Koch-Berger, who gave a quantum mechanical theory for thermal mixing. And the paper is very rigorous, but the highlight the highlight of the paper is basically one needs to have an asymmetric coupling network of the radicals in order to observe the thermal mixing. So if it is an asymmetric coupling network, the EPR line shape would also be asymmetric. If we go back to like old papers, the classical thermal mixing paper, which are based on Provotorov's theory, there also most of the paper they talk about having an heterogeneity in the EPR line shape in order to have efficient thermal mixing. And one can understand why do we need asymmetry. We need asymmetry because in case of a coupled electron spins, if there is no asymmetry, if we irradiate microwave, then we will saturate the entire EPR line and we will not be able to generate a polarization difference between electron. Generating a difference is very important. So for that, having asymmetry is very important. So that's the first theoretical prediction. The other prediction, which was made very recently, was from the lab of Shimon Vega, and he and Daniela Goldfarb, they have introduced the concept of ELDO to the DNP community. So they predicted that if you have coupled electron spins, then for certain electron spins, there would be hyperpolarization in the electron experiment and in the ELDO experiments. Very recently, when I was there in a workshop in Brno, I met Nino, and he told me like, okay, yeah, this is there, and it can also be used for doing efficient spectroscopy. So there is a paper on it where using this feature of ELDOR, where there is hyperpolarization, one can know about the distance between the coupled copper spins. So we had these two guidelines, which we wanted to check with the EPR experiment. And in fact, like we were very happy to see that in case when there is thermal mixing, which is at high concentration of trityl radical, there is a large asymmetry in the EPR line shape. And as concentration increases, as this asymmetry increases, and also the thermal mixing efficiency increases. The other feature is hyperpolarization. So what I mean here is that typically if you do an ELDOR experiment, you would expect that you burn a hole by saturating the electrons. But if the coupling network is very strong, then we also create hyperpolarization, which can be understood if from a multi-energy level system. So, and we could very clearly see that in case of 40 millimolar of trityl, in addition to saturation, we also see hyperpolarization of the electron spins. So this is definitely a clear indication of thermal mixing from EPR detection point of view, something that NMR could not have predicted. With the establishment of a system of EPR experiment that can be used to detect DNP mechanism, we wanted to use this study and extend it to something that is very interesting. So in 2014, there was a paper that got a lot of attention. The paper came from the lab of Robert Griffin, who showed that in case of BDPA radical, there is, in addition to solid effect, there is an additional enhancement that comes at the center of the BDPA radical. BDPA in one sense is very similar to trital because that's also a narrow line radical. And using NMR experiment and some other analysis, it was predicted that most likely the operating mechanism is Overhauser. So that was the best prediction that could have been made at that time. But the paper also highlighted that it's not very clear, or it's not confirmed that it's Overhauser effect. There can be some different mechanism operating as well. This paper also was very important because I remember I showed you that we want to push the boundary of DNP towards a more prominent condition. This particular study showed that this particular system was basically a solution, showed to be a potential solution to achieve this. So for all that reason, this was very interesting. But then the mechanism, having an Overhauser mechanism, was always doubted. So we since had this this information about trital clustering and giving rise to thermal mixing. So we just wanted to test if something like that is possible or not. So Miranda Lee, who is a graduate student in our lab, she did all these studies and a very systematic studies, and she showed that in fact in case of BDPA, it's the thermal mixing DNP mechanism that's operative. We get very similar signature which we got in case of trital. So there was asymmetry in the EPR line shape, and there was hyperpolarization of the electron spins. So so this was something that that made us extremely happy about the kind of EPR detection that we could use efficiently to find the DNP mechanism. So using EPR experiment, we also got a set of information about the properties of the BDPA at high concentration, and we used those properties like the relaxation rate and the coupling network to do some simple quantum mechanical simulation. And we were very happy that we could model the thermal mixing line shape for a coupled electron spin system. So our conclusion from the study was it's the coupling network between the electron spins that determines the polarization gradient between the electron spins, and the shape of polarization gradient in turns determines the shape of thermal mixing DNP profile, and it can be absorptive in certain cases, or it can also be dispersive. So in case of BDPA, it happens to be absorptive. In case of trital, it happens to be dispersive. Right now, we don't have, like, we know like it's dependent on the coupling network, but we are not exactly very sure why a certain kind of coupling network gives rise to a certain polarization differential profile or the thermal mixing profile. So this is something that we would continue in future in our lab. So with this, I want to first acknowledge some people who were actively involved in the research that I showed you here. So basically, Sogi Han has been the main factor in all these projects. The idea was there already, and we were able to implement recently. Projects that I showed you here also involved Miranda and Tanuma, who played a very important role in all the experiments. And the setup that I used was developed by Khan Tagami, Sheetal Jain, and Ilya Kaminkar, who has now become a faculty in Tel Aviv University. And we got a lot of ideas by discussing with people at Weizmann, especially Shimon Vega, Daniela, Akiva, and Krishnendu, who was a postdoc back then in Weizmann when I visited there to learn something about DNP. So with that, I would conclude. I hope that I have been able to convince you that EPR detection is very important if we want to decipher the DNP mechanism, especially knowing about the E coupling network and the polarization gradient under microwave saturation irradiation condition is very important. And I have hope that by combining EPR and NMR, we can bring another renaissance in the DNP research. So with that, I'd be very happy to take any question. And like before I take question, I want to pitch for the Twitter page that we have for our lab. So at Songi Khan Lab, you can follow us. If you want, you can also follow me at Asif Equbal 313. Thank you.
Thank you very much, Asif. So if you have questions,